move now to Paul Burton.
Yep.
I think it's a double act, isn't it? This?
Well, it's meant to be. That's one of the first things I was going to say. It's sadly not going to be a double act. Okay, great. So, so this is meant to be me and Madeline, both from the DCK Research Group in Bristol. Unfortunately, Madeline's developed a migraine this afternoon, has had to go off. And so you're going to have the first experience of any audience ever of me presenting some social science. So I hope that it goes well. Okay, so, and now the other thing to say before I start is that this is a section on genomics and governance, and I am presenting a case study for the 1958 Birth Cohort. But in fact, the governance of straight genomics is actually fairly straightforward in the 58 cohort, with the exception of the issue of feedback of results. And therefore, what I'm actually talking about is a bit more broadly how the governance works when we have an interface between genomics and other parts of the bioresource. Okay, so 1958 Birth Cohort started life as the National Child Development Study in 1958, and basically what happened in the week from the 3rd to the 9th of March in that year, they tried to recruit all children born in Great Britain. So it's not United Kingdom, it's Great Britain. Northern Ireland wasn't involved. And basically this was funded by the National Birthday Trust Fund. It was a study of perinatal mortality. And at the end of the day, they ended up recruiting just over about nearly 17,500 people. 7 years later, this became a longitudinal cohort where, with funding for the Department of Education and Science, they then did a school-based assessment, which ended up recruiting 15,000 people, re-recruiting 15,000 of those people at the age of 7. Then over the next, there were 5 further sweeps between 1969 at age 11 up to year 2000 at age 42, funded by a variety of groups and ending up particularly with the ESRC. Then the most important bit from my perspective is in 2002-2003, the Biomedical Sweep took place, and this is the basis of the biomedical resource of the 58 cohort that I have responsibility for overseeing, and I'm going to say more about that in a minute. Since then, there have been 3 non-biomedical further sweeps from 2004 up to 2013. Okay, so what was the Biomedical Sweep? So it was carried out 2002-2003, when they were aged 44 to 45, and it was funded under the MRC's Health of the Public initiative. There were 3 PIs initially, David Strachan from St George's, Chris Power from Institute for Child Health, and initially John Bunner from the Centre for Longitudinal Studies, but his role was passed on to Heather Joshi fairly soon after the study was started. For the Biomedical Suite, there were 16,000 people that in the cohort that they targeted, 12,000 were able to be contacted, and 9,400 were actually interviewed. And it's probably pretty obvious, but the main primary objective was stated to be to obtain critical biomedical information via questionnaires, physical measures, and biospecimen collection, which included blood, urine, and saliva. And the aim was to examine how developmental, lifestyle, and environmental factors act throughout the lifespan to influence current ill health and physiological and psychological function into the middle age. In addition to the funding from the MRC, there was also some funding from Wellcome Trust, which was very important because it allowed, there were about 7,500 people who had blood taken for DNA extraction. But in addition, they produced about the same number of lymphocyte cell lines. So basically we had a permanent resource for DNA, which was then a major use later on. And I'll say about that in a second. And at this point, it was clear that we needed some form of access mechanism for both data and biosamples. Now, initially, a fairly conventional PI-centered mechanism was set up, which ran for, I can't remember exactly how long, it was something like, it was either 4 or 5 years, and basically increasingly over time, it became more and more problematic. People were feeling that it was taking much too long for the applications to be processed, and there became a sort of an impression, which I think you can argue was an incorrect impression, but nevertheless it was a widespread impression that at least one of the PIs was deliberately trying to block access to the data and samples, and this became hugely problematic. So in about 2007, MRC took over the data access process themselves, and that also didn't work. There, the problem was that in order to be a sensible data access mechanism, you required quite a lot of knowledge about what was actually happening in the study. And what it turned out was that all the difficult issues that take time ended up having to be passed down to the PIs anyway. So basically, having it sitting in the MRC wasn't a particularly sensible way forward. So basically, in 2008-9, the MRC and Wellcome Trust decided that given that the infrastructure, so the people, systems, and processes that you need to get streamlined access to data and biosamples from the 58 cohort was pretty extensive. It needed quite a lot of these. They decided to directly fund an access infrastructure grant. Initially, it was just a one-off small grant for a year. And then from 2011, they then funded a 42-month grant. Initially went to me at University of Leicester and Sue Ring and George Davies Smith in Bristol. And then in 2013, myself and Madeline basically moved to Bristol. And at that point, the full grant then moved to Bristol. Now, at the same time as setting up that infrastructure, so actually deliberately funding a proper infrastructure for access, the ESRC jointly with MRC and Wellcome Trust decided that given that that this investment was taking place in a proper infrastructure, why don't we try using it to have a broader remit than just the 58 cohort? And so we became the Access Committee for CLS cohorts with responsibility for the biomedical bits of the 1958, 1970 Millennium cohorts. We then later took on responsibility for UK twins, and Massimo, who's here, is one of the technical group on the Oversight Committee. And very recently, we've taken on additional responsibility for the Next Step study, which is a new CLS cohort, which is originally was called Longitudinal Study of Young People in England. All right, so how does application work? So basically, first of all, there's an application form that's downloadable from the web. So applicants fill that in. They send it to the Secretariat, and the first thing that happens is they check that that's complete. And then the next thing happens is, provided that is complete, it then goes to a technical review committee, which look for major scientific, practical, or governance issues. And if it passes through that, it then goes to the full access committee, which has got 8 full voting members. But critically, and I think this is really important, the technical review team also sit on the access committee. So although they don't vote, they can provide very important background information if there's discussions about the science. And in addition, we have representatives from all 3 funders, but they are non-voting. Now, critically, basically the 2 issues, there's streamlining and there's making sure that you're protecting the cohort in ways that I'll say in a minute. And in order to ensure streamlining, what we do is as soon as we receive an application, first of all, if it's not complete, we immediately send it back and say, please complete it. If you can get it back to us in a couple of days, then it can just progress. Similarly, if when the technical review committee look at it, there's something remediable in terms of a big problem in what they're doing, then we get them to do that. And basically, if people can get an application to us at least 1 week ahead of the next meeting with all those problems resolved, then we will then review them at the next meeting. And so that seems to generally work pretty efficiently. A key issue which has really only recently in the last year been properly formalised is that we've developed an appeals process now. So basically, if there is any formal concern about a decision, then it would be referred up to the Centre for Longitudinal Studies Strategic Advisory Board, so no appeal has ever been requested to date. Right, so how do we judge the applications? Well, the critical issue is that we're judging a number of things that don't include the science. So basically, first of all, has the application been submitted by bona fide researchers? Secondly, does the application violate or potentially violate any of the ethical permissions or other consent forms? And this is where the potential for issues about feedback of clinically relevant results come in, because we have an absolute ban on returning any clinical results from genomic data, and yet if we had a potentially clinically relevant thing, how do we deal with it? So that's something we would need to deal with at the time. Is there any risk that the application could lead to identification of individual cohort members? Is there a risk of upsetting or alienating cohort members or reducing their willingness to stay in the cohort? Does the application address topics that fall within the acknowledged remit of the project? And it's a very wide remit. It's effectively a broad consent procedure. And then finally, there's a final question: does the application require access to an infinite resource, so the data or the cell lines, or a finite resource, so whole blood extracted DNA, blood, saliva, or urine? Critically, we only assess science in some specific cases. First of all, if the application is for a finite resource, because then it's competition with other potential scientific applications. Secondly, if there's a scientific issue impinging on other criteria. And so, for example, we actually had a meeting yesterday, and at that meeting we had quite a discussion relating to the salivary cortisol levels, and that was because we were dealing with a particular variant which had been suggested might be related to adrenal hyperplasia, and the question is whether or not we needed to be worried about that as a feedback issue. There's the other situation where we ask for it is if there's a request for a very large range of data and biosamples. So if people start asking for a significant proportion of the underlying resource, then we really check, do they really need this for what they're asking to do, or are they just taking things just in case they might need them? And then the other thing that happens all the time is that the technical review team does assess the science, not in terms of saying, is this a good bit of science or a bad bit of science, but in terms of literally, is this what's proposed practicable at all? So, for example, if they put an application that's only going to produce 2 cases in the whole of the 1958 birth cohort, that may not be a good idea. Equally, we had one application fairly recently for a study that actually required children, so they didn't understand that it was an adult cohort now. So those sorts of things that they get picked up. All right, some additional information. Over the time that we've been working, there have been effectively 3 waves of different types of application. So that should really be from 04 to 06. We were getting applications primarily for blood, largely from the DNA from the lymphocyte cell lines to do genotyping. So basically, that was the period when a large amount of genotyping was done. Between 7 and 10, a lot of those genotypes were then being used, often as control series for other UK case series. And then since 2011, we've increasingly moved over primarily to dealing with linked genotype-phenotype applications. Now, these are important because they potentially introduce more serious concerns for A, identification, and B, for inference then based on those identification. So basically, one of the things we found is that as we've increasingly moved to be dealing with those, we've had to increasingly have a lot more discussion about things on the committee. So this is important for new studies starting up, like, say, Understanding Society. At the moment, it's at the early phase. Later on, it may have to do a lot more discussion. The study has been very productive. So if we just look at the Biomedical Sweep stuff, then between 2004 and 2013, there were a total of 632 peer-reviewed papers that we identified when we did our research fish last year. Perhaps the most, well, in my view, definitely the most important was the Wellcome Trust Case Control Consortium, where we provided half the controls, but the very wide range of other papers as well. And the other thing that's really important is the need for strategic interactions. Basically, everybody involved with data access knows that there are a lot of strategic links that we need to take advantage of when we can. So, for example, the ACCC only oversees applications involving biosamples, requiring a linkage of genotype to phenotype, and challenging applications if you're only asking for phenotype or genotype alone. Otherwise, if you're only asking for basic genotype, it now gets assessed by the Sanger Data Access Committee. Basic phenotype alone gets assessed by the Centre for Longitudinal Studies. We have links for biosample storage we mainly hold it in Bristol and Alspac, but UK Biobank stores some of our biosamples as an inactive backup, and the cell cultures are held in an inactive backup at ECAC in Porton Down. The Centre for Longitudinal Study acts not only as our appeals board, the strategic advisory board of that, not only our appeals board, but also one of the key things that we found in discussion, and in fact we actually specifically discussed this at our meeting yesterday as to whether or not it was sensible. We believe that you need, in order to keep up with the horizon scanning and understanding, developing new issues as they go along, you actually do have to spend a substantive amount of time in these access committees actually discussing those things properly. It's very tempting to just keep making boom, boom, boom decisions on studies, but over time you're building up issues and lack of understanding. I think it is very important that that is recognised, that that's part of the time required of an access committee. One of the things that earlier on today, we were talking about big biobanking consortia and whether we're setting things up right for that. In fact, we've had opportunities to do that within 58 cohorts. So there's a number of major consortia that we provide data and samples to. And the other thing is that we've shared our experience with other major studies. So, for example, Paul Robson's here from the Alberta Tomorrow project, and we've had discussions with them when they were setting up their access committee. Have I got any time left? Not much. Okay, maybe what I suggest is rather than presenting the social science badly, why don't I skip that? Apologies to Madeline. Yes, okay, all right, I'm being a chicken. Okay, just to finish off then, the last slide. So data access is only one part of governance, or governance around data access is only one part, but it is a major challenge. First of all, multiple funders are involved, and one of the things I'd like to emphasise is at the end of the day, it's almost the funders have the biggest load on them in relation to that. For the scientists, if we were told, sorry, you're not going to not provide any more data access again in the future, all right, it would be, if you believed in data access, that'd be a problem, but you would be able to continue doing your own research. But the whole point for the funders is that if they don't deal with data access correctly, they're going to be making inefficient use of important resources, and both nationally and within individual funders, there'll be a problem. The problems are compounded by the fact that there are multiple studies involved, and what we find is that there are commonalities between some studies and heterogeneity between studies. So in one sense, we can't discuss everything in one national data access committee because there's so many specific issues that we would have to do something at the local study. On the other hand, if we try and do everything at the local study level, then we're wasting a lot of time. So basically, this is something that needs to be dealt with. And this is again compounded by the fact there are multiple stakeholders, sometimes with conflicting desires about things in terms of the balance between streamlined access and security, for example. Finally, these things, it is extremely resource demanding. We have to supply mechanisms that give streamlined release of data to people who've been assessed as being bona fide applicants. We have to ensure robust security constantly. And then there's this issue of the need for extensive strategic discussion and planning, following on from horizon scanning to identify, predict, and manage challenges as they arise. Okay.
Thanks very much, Paul.